Good afternoon, and thank you very much to the organisers for inviting us to speak about the translational cut research that we're doing here in Manchester. So I'm Alicia, I'm a clinical fellow undertaking a PhD at the Cancer Research UK Manchester Institute, headed up by Professor Caroline Dive at the Cancer Biomarker Centre. And my clinical supervisor is Dr. Natalie Cook, who's here in the audience today, and she's an experimental cancer medicine consultant at the Christie Hospital. And it's really these two people that have been the driving force to build the foundations of our translational cut research here in Manchester. And my PhD started in 2017, at which point the CRUK Institute was actually just a building away from the Christie, and it was very easy to get samples to and from and sort of collaborate. But due to a fire in 2017, the Institute's now moved to the lovely picturesque rural parts of Cheshire that you see, but it means that we're now geographically away from the Christie, which has had its challenges, but we adapted, and we're still doing great translational cut research despite this. So the overview of my talk really is to first introduce the diagnostic and research challenges of cut and the questions that we have decided to focus on. I'll introduce my PhD and the role of liquid biopsies. I'll give an overview of the CUP COMP trial, which is a national trial led by Natalie, due to open in the next few weeks. And I want to just introduce a new project we're starting, the CUP Peace project, and finish with an overview of the Manchester Research Network, which is actually more of a national network now, and talk about some of the future ambitions. So, as we all know, there are lots of diagnostic challenges in CUP, and we really feel it's these diagnostic challenges that give us research opportunities to make clinically meaningful differences for patients with CUP. It's a lot like John Simmons mentioned at the end of his talk. It's really focusing on the things that can improve clinical outcomes for patients. So I'm just going to go over some of the common research questions that have tried to be addressed in CUP, and how we might use those to focus our research. So the first one is can we determine tissue of origin in CUP by molecular profiling? And you'll hear later on this afternoon about the trials that have tried to address this question. I think overall we probably can predict tissue of origin. The big question is, does it improve outcomes, and how do we prove that it does improve outcomes within the limitations of the research? The second research question is there a unique common biology for all CUP tumours? And I don't think we've really described that to date from the evidence. And we know that there's lots of heterogeneity within CUP cohorts. And I really feel that there's more differences than there are similarities in our CUP cohorts. The third question is, can we detect molecular driver mutations to direct therapy? And we know we can in known tumour types, and we have seen this in CUP patients. We can detect molecular driver mutations. They can enter onto phase one trials. But there is a real challenge with CUP, and that is we know that some of these therapies that act on these driver mutations are tumour type specific, and their efficacy is uncertain without a tissue of origin. And also, most of the access to targeted therapies is based on tissue type. So how do we get access to these therapies outside of clinical trials, even if these patients have driver mutations? And here at Manchester, we really feel that it's both of these tissue of origin and mutational profiling together that will be important for treatment decision-making and access to appropriate therapy. And the final question is there an alternative to tissue biopsy? We know that tissue biopsies are sparse in CUP patients. Often they've had multiple immunohistochemistry stains for diagnostic purposes. And actually, we need good quality and quantity of DNA for molecular profiling. So our approach at Manchester is to try to use liquid biopsies as an alternative to tissue biopsies, and that's the focus of my PhD. So my PhD hypothesis is looking at the role of liquid biopsies and can they be used to improve diagnostics and management stratification in patients with cup tumours. Currently, patients with confirmed cup fall into either a favourable or unfavourable subset. We know that patients with favourable cup have tumours that mimic that of known tumour types. They can go on to have molecular profiling as appropriate for that tumour type and have site-specific therapy, be that immunotherapy, chemotherapy, or targeted therapy. We know these patients generally do better than the unfavourable subsets. But unfortunately, the majority of our cup cohorts fall within the unfavourable subset, and these patients currently are offered palliative chemotherapy or best supportive care. And they're really treated as a homogeneous group of patients, despite the fact that we know there is clinical, pathological, and molecular heterogeneity within this group. We propose that a liquid biopsy, predominantly multi-omics profiling of cfDNA, will enable us to predict tissue of origin and look for predictive biomarkers, which will help stratify our patients into more favourable subtypes, into more disease-specific pathways that we know have better site-directed, site-specific therapies. And this is the main aims of my PhD, to predict tissue of origin, identify predictive biomarkers, and really start to characterise these more favourable subtypes. So what is a liquid biopsy? Well, we predominantly deal with blood, but it can be any fluid from the patient. And we know within the blood that we find cancer patients can have circulating tumour cells and also circulating free DNA, for which a proportion of this is derived from the tumour. And there's multiple analysis that we can do on the CTCs or the DNA. In our lab, we're able to isolate and characterize CTCs. We can also implant these CTCs into mouse models and develop and produce CTC-derived xenografts. The circulating free DNA can be extracted. We can do sequencing to look at copy number alterations and point mutations, and also look at epigenetic modifications. And the focus of my PhD has been looking at epigenetic modifications, predominantly DNA methylation, which you'll hear about from Manel Estella, who's really done the groundwork in proving that DNA methylation is both tissue and tumour specific and actually enables tumour type prediction from tumour tissue. So we want to do something similar in the cfDNA, and we've developed a novel methylation protocol enabling us to perform whole genome methylation evaluation from the cfDNA of these patients. So the next steps is to see if we can distinguish the tumour of origin from these methylation patterns. So next we ask, what about circulating tumour cells? So these are rare cells in amongst millions of red and white blood cells. So it really is finding a needle in a haystack. And there's very limited published evidence of CTCs in cup. There are two publications to date that have identified CTCs in cup using a cell search technology which requires the CTCs to carry an epithelial marker for identification. And what we know is that often CTCs lose this epithelial marker, and also we don't know in cup how many of those patients will actually have CTCs that carry this marker. So here in Manchester, we have the advantage of multiple different CTC technologies within our laboratory. So we've used a technology that doesn't require this epithelial marker, but actually selects based on the size of the cell rather than any antibody it may carry. So CTCs are generally larger than white and red blood cells, and this Parsortix method is able to capture these circulating tumour cells. We can then stain and isolate for these CTCs on the DEP array and then use downstream molecular characterization to confirm that they're CTCs. So we use markers of cytokeratin, an epithelial marker. We use CD45, which is a white blood cell marker, and vermentin, which is a mesenchymal marker. And this is able to identify suspected CTCs and any white blood cells we may have carried over. And this works really in its infancy in CUP and in by project, but we are starting to see some of these suspected CTCs in CUP patients, and we want to go on to molecularly characterize them to see what these cells actually look like. So that's a little sort of flavour of the projects within my PhD, and I want to move on to talk about the CUP-COMP trial, which is led by Natalie Cook and is a national initiative to assess and compare the genomic sequencing in tissue and blood for the diagnosis and treatment stratification of patients with CUP. And this is a really exciting project that brings together commercial entities alongside academia across 7 NHS sites, recruiting patients in real time to get tissue and blood molecular profiling and feed this into a virtual CUP molecular tumour board to make real-time clinical decisions. It's really exciting that these patients will have access to molecular profiling, but the really important part of this project is it's going to be a platform to secure further funding and expand the initiative. And from a research perspective, we'll be able to build up a biobank of samples with robust clinical data attached to it for future research. And finally, I'd like to tell you about our CUP Peace project that's recently had approval. The Peace initiative was started by Charlie Swanton at UCL, initially to look at lung cancer tumour evolution, but has been opened up to other disease types, and we've had our CUP Peace project approved, and this will give us access to post-mortem tissue from multiple tumour sites from patients with CUP, and potentially be able to find the primary post-mortem. We really feel that this project can answer some of the key biological questions into the metastatic processes in CUP. This diagram illustrates really nicely the fundamentally unanswered questions into the biology of CUP that are still debated. Is it that there's a small undetectable primary tumour that seeds an aggressive metastatic phenotype? Is there primary tumour regression and that's why we can't find a primary tumour? Or is it that no tumour exists or ever existed, and actually this metastatic phenotype arises from a pre-malignant stem-like cell? And we feel that CUP Peace may be a great way to try to get some of the answers to these questions. And we want to do that by evaluating the immune microenvironment of the metastasis and hopefully compare this to a primary if it's found, investigate the intertumour and intratumour heterogeneity, and also track the molecular tumour evolution of these tumours to try to answer some of these questions. My final slide is an overview of what we've built in the last sort of 4 years. So the humble beginnings of my CUP PhD and the CUP Biobank back in 2017, we've actually built a network of partnerships and collaborations that mean we can do more and more valuable research for our CUP patients. And looking to the future, we hope to be able to collaborate with people like Richard Tothill in Australia and Manela Stella in Barcelona, and really build on these foundations to join them in their sort of as international leaders in CUP research. So that's our ambition. And finally, I'd just like to finish by thanking all the individuals, all the companies, all the funders, and all the collaborators that really have made this work possible. My PhD is funded by the Manchester Biomedical Research Centre and CRUK and the CUP Foundation. The driving force behind the research is Dr. Natalie Cook and Professor Caroline Dive, and their support has been invaluable during my PhD. And really special thanks to Roche, who have collaborated on Cup Comp and other projects and really been supportive in our cup research. And thanks to the Cup Foundation and John Simmons, who was there when we were just starting out, giving his support and advice on how to sort of make our way in the cup research field. So I also need to thank the patients and their families, for which without them, none of this research would be possible. And I thank you for listening, and I'm happy to take any questions.